And now we're moving up to the heart with Laksshman Sundaram from PhD student at Stanford University in computer science. So you're on.
Thank you. Can you see my slides well?
Yeah, perfect.
Awesome. Thank you. Thanks a lot for giving the opportunity to present our work in the meeting. So I'm Laksshman Sundaram. I'm a Stanford PhD student working with Marshall Kondaji and William Greenleaf's lab at Stanford. So one of the questions that we wanted to answer in the lab is that the human heart is the first organ to form, and the right functioning or the right formation of the human heart is very important for its own functionality, but it is also very important for like all of the subsequent organogenesis that's going to happen. So one thing that we wanted to understand is what is the process that is driving the regulatory like heterogeneity of the human cardiogenesis process, because congenital heart disease is found in more than 1% of all live births. So really understanding how the regulatory changes happen over a period of time during the developmental window can really help us understand what are the potential downstream regulatory changes that could eventually cause like congenital heart disorders in these kids. So to do this, we sampled 3 hearts, 3 human fetal hearts, one 6-week post-conception, the other 8-week post-conception, and then finally 19-week post-conception. And we did like 10x based single cell attack sequencing of these 3 different hearts. And one thing that we could already say from each of the individual time points is that as we go through the time points, we actually have like more cellular heterogeneity that we actually could observe with these hearts. And all of this data here is being generated by Mo, who's a very talented wet lab experimental grad student at Stanford as well. So one thing is to look at these cells as like just discrete time cells, but then the other way of looking at it is now putting all of this together and then looking at like a developmental trajectory or a developmental landscape of these different cells. And the first thing that we try to do with such an atlas is that we try to annotate like cell types of the heart. And what we actually see is like a very rich cell state annotations that is possible using like chromatin aggregated signal of like marker genes. So what we roughly have is like the myocardium cells, which form the muscular part of the heart over here with all the cardiomyocytes. And then here are the epicardium cells, which form the outer layer and also the vascular smooth muscles. And then we finally have the endocardium and the endothelium cells, which are like arteries, capillaries, veins of the heart. So having had such an atlas, the next thing that we wanted to understand is like, we wanted to really resolve like per base pair specificity of the DNA sequence in actually driving these chromatin signal. So what we do is we train a sequence to the TN5 insertions from the attack-seq model for each of the cell types separately, which basically is like a text-to-speech converter where we take in the sequence and then predict the TN5 insertions. And one thing that we could also do with these models is by back propagating through the layers, we could actually get to the input sequence and then really understand like what are the drivers of these, accessibility. So just diving in directly into the disease genomics, this is an example of a point de novo mutation at the Jared locus, which is a gene implicated in like congenital heart disease. So what we actually see is when we actually have a G to C change that is over here, the G is turning into a C, the neural network model is actually predicting that a SOX13 binding site that is predicted to be active in the arteries is actually being disrupted in the case of the congenital heart disease. And you could also see that this gene is specifically expressed only in the cell type. And then we could also have many more such examples of like point mutations that really cause a phenotype. And just skipping ahead, one thing we also did is we made these edits in like iPSC-derived endothelium cells and iPSC-derived neural crest cells, and you can actually observe the downstream downregulation of the genes that we implicate to be like affected in congenital heart disease. Just a quick point, there was a question on like comparison of, in vitro derived cell types to fetal cell types. Please check out the poster. We have like a version of such an analysis. And to conclude, I would like to thank all the collaborators, Anshul, Will, my advisors, Mo, Tom, Kevin, and all of the collaborators that made this project possible. Thank you.
Thanks a lot, Laksshman.